Well, good morning, good afternoon and good evening, everyone. Welcome to the Marine Biological Association of the UK. We're delighted to host this event for the CPR 90th from our world-class facilities here in Plymouth. We're really sorry that you can't join us in person during current COVID times, but obviously we look forward to welcoming you back here soon. In case you aren't aware, here at the MBA, we offer a variety of training and events opportunities available to our staff members and non-members. So please do keep an eye out on your membership bulletins, website and social media platforms. And we'll also be sharing some of the details on these and the holding slides in the break time, so please take a look. I should say that whilst we're holding this on Zoom, we will be live streaming to a number of social media platforms as well. The sessions will also be uploaded to the MBA YouTube channel in the coming days, so you can watch this at a later date if you cannot stay for the whole meeting, and perhaps at a more sociable time for some of our overseas viewers. I'd just like to say a quick thank you for all the work of the organising committee in getting this sorted, and to all the speakers and to all our attendees. Thank you for taking the time to come along to celebrate virtually with us. So as you all know, the reason why we're here is the 90th anniversary of the first tow of the CPR by Sir Alistair from the SS Albatross in the North Sea. I can't imagine that Sir Alistair could have possibly thought we'd still be here all those years later, reaching over 7 million miles of tow, a Guinness World Record, surviving a world war, a global pandemic, and more importantly, thankfully still receiving funding to run our unique time series. His ideas about helping herring fishermen have grown dramatically over the decades, and today's talk should hopefully give you a good idea about some of the areas of our work. It's truly a testament to his design and vision, as well as a dedication to all the staff who've been involved over the years, that the survey has been such a success. But perhaps more importantly, we couldn't have achieved what we have without the support of the shipping industry. We're truly indebted to everyone involved, who braved some truly awful conditions at times to tow for us throughout the world's oceans each and every month. So here today, we're going to celebrate our 90th with a series of talks, and they're going to be in 4 sessions. Firstly, we felt it was important to look back to the past to see how we arrived here. So we have talks on the CPR history and development, the survey, and importantly, a talk about why we should take care about plankton and taxonomy. For the first session, our opening talk will be by Professor John Spicer, and that's entitled When the Wanderer Met the Hardy Baby. John will also be acting as the first session chair. Then we'll have Professor Chris Reid at the MBA and a former director of SAFOS, who'll give a talk on 90 years of the CPR survey, a potted history. Next, it will be Dr. Alistair Lindley and Tony John, a couple of happy retirees formerly from SAFOS, who'll give a talk entitled Conversations Around the Microscope. And lastly, in the first session, Marianne Wootton, who's the senior analyst here at the CPR survey, a talk on what are plankton, what is taxonomy, and why should we care. Our second session will concentrate on how we have grown from that first tow in the North Sea to a global community of CPR surveys, with talks showcasing research from around the world. Thirdly, we wanted to highlight the impact and the applications of our CPR data. Yes, we collect plankton, but those plankton sit at the base of the marine food web, and those resulting data can be applied to higher trophic levels such as fish and seabirds and new societal issues. And finally, we take a look to the future. What next for the CPR survey? All of you have been involved with our work know that we constantly strive to keep our work relevant, always looking for how we can apply our data, our samples, and our network to future issues. With climate change, always so important to us, being very high on the agenda now, it's timely we have this opportunity to share our work with you. We'll also be sharing a short survey after the event, and we'll give the opportunity to say where you see the CPR survey and research going in the next decades. So a couple of details about how everything's going to work. Each talk is going to be around about 15 minutes long, and most of them have been pre-recorded to help with the timings. And each session will have 4 talks. There'll be a break of 15 minutes between sessions 1 and 2, and between 3 and 4, and a longer 30-minute break for lunch between 2 and 3. Whilst we're here virtually, we do want to make this day as interactive as possible with time for questions. So I'd like to introduce Maya Plass, our head of comms, and she'll quickly explain how that will work. But I'd also like to say that, any of us will be happy to be contacted by email if you want to discuss any other sort of work or collaborations or access to our data set.
Lovely, David. Thank you very much. Before you disappear, can I just ask you, you're doing a very good job of modelling our beautiful T-shirts. Could you just stand up and show our lungs of the sea, the plankton? Nicely done. Thank you, David. So these are our T-Mill T-shirts. So you can get these from our T-Mill website, which I'm sure one of the people behind the scenes will be able to share the link to. So thank you for that, David. And welcome to the CP90 event. It's been an amazing year, and there seems, as David has said, to be endless celebrations for CPR90 this year. And it is a shame not to have you attend physically with us here, but the wonder of virtual events is that we're able to have you attend from all over the world. So we've already had Karen say that she signed in from New Zealand. Sonia is in Canada, I think it is, at 2 a.m. So thank you for all you early birds joining with us. Our registered attendees are from Pakistan to Poland, Australia to Argentina, South Africa to Seychelles, and Singapore to Sweden. And I think that sort of represents that global reach of the CPR survey over the 90 years. So a very big welcome to you all. So just to explain the question and answer functions and how it works. So at the bottom of the screen, if you run your cursor along the bottom of the screen, you'll see a Q&A function. If you click on that Q&A function, you'll be able to ask your questions. And once they're in there, I'll be looking through those. And then at the end of each theme, I will ask those questions to the panel. The alternative, so that it makes it a little bit more interactive, is you should also have a raise hand function at the bottom there. If you raise your hand, that means that we can then allow your microphone to be switched on. Somebody's just said, don't forget the small island of Cyprus. Welcome to Cyprus as well. And if you switch your microphones on, you then are giving us permission to record you talking. Unfortunately, we don't have camera functions on due to consent, but it'd be great to have a bit of interaction should you want to ask questions through raising your hand and speaking verbally. That'd be fab. Behind the scenes, we've got Neri on social media, because we are sharing this on our social media platforms live on Facebook and YouTube. Welcome to our viewers on Facebook and YouTube. And if you've got any questions, feel free to pop them in the comments, and Neri will transfer them to the Q&A function here so that we can all see. Behind the scenes, we've also got David, Maz, Jennifer and Gemma, who can answer sort of more specific questions about CPR or any sort of particular contacts, but the general questions will be asked at the end of the panel, and hopefully we'll be able to get through all of those. As David also mentioned, we've got, so I've got somebody else saying, John Kitchener from the Australian Antarctic Division here, 7:15 p.m, hope to last until midnight. Strong coffees are recommended there. Lovely to have you with us. And then just to remind anybody that's joined us slightly later, we have a survey that we're going to be sharing a link for. It's a very short survey, but it does really help us understand our audience, see what's working well, see how we could improve for the next event, the 100th event, which is Monday, the 15th of September, 2031, should you want to put that in your diaries. So please do go straight to that link if you could at the end of this day and fill that form in. That would be really helpful for us. And before I pass back to David, I just wanted to introduce our special guest next. So I wondered if the special guest could please turn their camera on for me. Hopefully we can spotlight that for you as well. I don't know if you can see that. So we have a rather special cake, and we are very sorry not to be able to share this with you. Are you all able to see the cake? Hopefully you can. So this is a rather lovely cake with a cake topping, which is of the SS Albatross, which David mentioned. So it was on this day in 1931 that the first CPR was towed from Hull in the northeast of England to Bremen in Germany, so 90 years later. So this cake represents all those years of tows. It's got a raspberry and cherry sponge inside. As I said, we are very sorry that you can't share that with us. I hope your taste buds are tingling now, and we'll let you know how delicious it is. So thanks you to Conradi Cakes for delivering that. It was a very petrifying drive to the building to make sure that it didn't fall apart. So I shall now pass you back to David. Hope you have a wonderful day, and thank you for joining us from all over the world.
Thank you, Maya. So right, well, to kick off, it gives me great pleasure to introduce Professor John Spicer from the University of Plymouth. John has been a great supporter of the CPR survey over the years, and in fact opened at the 80th anniversary as well. So I'm hoping John has now penciled in for his diary for the 100th. John's also going to act as the chair, as I said, for the first session. So John, thank you, over to you.
Good morning, everyone. Well, it's good morning where I am in St Ives, where I've been doing my impression of the CPR this morning out swimming and not closing my mouth properly. A great kick-off to this history and natural history. A personal history, a history of the CPR, and an introduction to plankton. What more could you want from such a celebration? Three great speakers, and you've got me starting. And I'm going to address something which is something that I quite like, riddles. It's a bit of a riddle for you as well. I'm going to share my screen, so just give me a second to get sorted here. And around this riddle, if you want to put it like that, hold on, I'll just get rid of me. When the wanderer met Hardy's baby, now if you looked at that, first of all, you might have thought, what on earth were you coming to? I'm hoping by the end of my talk, which should introduce the other three, you'll have a much better idea of what the wanderer is and what Hardy's baby is as well. So let's kick off. So here's Kate, who's in a shop called The Green Space in Plymouth, and she's holding up a picture of a piece of plankton done by Debbie Mason. All round about we have fish and whales and everything else, but people here are buying images done by Debbie of plankton. I talked to some of them at the shop because my wife runs the shop, and they have no idea what they're buying. They just think they're beautiful. They just look and they still have no real idea what these creatures are. And when I sort of said, what this was, they were sort of fascinated, but clearly had not really thought about it. It's interesting, we've still got an awful long way to go to teach people about what these tiny plants, animals, algae in the ocean actually are. And yet there's been a really good history of public communication. Alistair Hardy, who gives his name to the foundation, put together what was originally supposed to be one book, a book on marine biology, but in fact it was just far too much for one book. And in the New Naturalist series, a series which shows the natural history of the UK and its waters, there are two books. Two books with the same sort of title, The Open Sea, Its Natural History. But notice the first part is entirely about plankton. The World of Plankton, published in 1956. Absolutely amazing book, and we'll say a bit more about that in a second. But notice, this is the natural history of the sea, and yet the part two is not just about fish, but it's also about fisheries. Because straight off the bat, Alistair Hardy and everyone who has worked in marine studies, certainly over the last 100 odd years, has realised that the natural history of the sea is, in one sense, a little bit unnatural now because we've made such a large impact on what's there. Fascinating set of books. If you haven't read them, if you've heard about them before, great, that's wonderful and you can reminisce. If you haven't, go and buy them. Particularly if you can get the original version, because you can see the incredible paintings and drawings that Hardy illustrated his work with. Incredible set of books. Here's the page which introduces the plankton. Now, of course, I don't want to steal any of Marianne's thunder, but here is Hardy starting off trying to tell everyone and anyone, as well as marine biology students, what plankton is. And it's interesting that when he was at university, he was very, very much into the idea of agriculture and helping and sort of this idea of doing good. And even in this book on plankton, he starts to be a marine biologist. He starts off with this, let us suppose for a moment that the herring is not a fish, but a land animal. And he starts painting this idea of, if you could look at the sea, the amount of what he calls plant material that's there, that's the basis of all the life. If it was on land, it would look incredible. And he uses amazing pictures to try and get across to folk what is actually in the sea, and in particular, what plankton looks like. And he gives in the next page a really lovely definition of plankton. And he explains it's from the Greek word planktos, which means the wanderer. So, and he gives a beautiful little piece. I won't ruin it for you. Go and read it yourself. A beautiful little piece when he's saying, yeah, but the Greek it's not just one word, it gives a whole feeling. And he goes into this idea of the feeling of the wanderer not being able to look after yourself, being passively dispersed, all this sort of stuff. Beautiful to read. Go and read it after the conference, of course. Hardy himself got involved in the love of plankton, really, when he was at Naples. He won a scholarship to Naples, and he says, and I don't exactly know why, but he says in one of his diaries that he was fascinated by marine plankton. 1921, with his scholarship over, he gets a chance to actually take part in plankton research. Starts on the 1st of August, assistant naturalist at the Fisheries Laboratory in Lowestoft. And his job is involving plankton. How did it come about? Because in the 1920s, there was a report in the British media that the French were spotting pilchards in the Bay of Biscay. Therefore, questions were asked in the House of Commons. Why was Britain not doing such surveys to help her fisheries? And so that's what happened. A research vessel, George Bligh, operating out of Lowestoft, was, if you like, commissioned to try and work out just what is the natural history of herring. And there was 4 or 5 people who were dedicated to this, one of them being Hardy, and his job was to study the feeding of the herring. Now, not just as its adult stage, but from its young stages as well, and in particular in relation to the plankton that occurred in the North Sea. And the picture that you can see here is actually from Hardy's book on the plankton, and it's an image which has been used through ecology texts for a long, long time, looking at the food web behind this particular fish species. It's classic. Even now, it's classic. Now, why tell you about that? Because this is 1921, right? This is 10 years before the first deployment of the CPR officially. And yet what I want to put to you is that the birth for the idea of the CPR and why the CPR came about, I think, is because of something that happened, something serendipitous that happened when Hardy was engaged on board the George Bligh. So here's the George Bligh. One of the main researchers, William Wallace, not the great Scotsman who shouted freedom and all stuff like that, but someone who was involved in asking where the young herring actually were. That was his question. He normally looked after the cruises, but this particular time he was ill, and there was a cruise into the southern bight of the North Sea, and Hardy was asked to sort of head it up. Efficiently, yeah, very efficiently. In fact, they finished the cruise a day early, which was incredible, really. Well, a day to spare. Day early is perhaps the wrong way to put it. But with that day to spare, Hardy says, well, let's go back to the part where we caught the most fish and just actually see what happens when we sample again. Because it was apparently it was an incredible catch of herring. So they went back to this particular station where they were looking for the herring. They'd found a lot. And he repeated the same process 4 times, looking for young fish at the surface, mid-water, and at the bottom. Repeated the sampling process 4 times. And what they found in that one day was that the difference in the numbers caught in the 4 repeated samples was greater than the entire variation in the numbers at all the other stations. It was also the same for the sand eels and the plankton. Now, clearly, that must have been really disheartening, because how can you say anything if there's no repeatability? And of course, at the centre of this is the whole ecological conundrum of how do you deal with patchiness? And it was in that 1921-22 period that Hardy set about thinking about how on earth can you actually quantify these planktonic creatures? How can you account for patchiness? And that's where the idea for the CPR really sort of came from. Here's a picture from Chris's paper from 2003, but the actual caption I've given is from Hardy's own book, Great Waters, Proud Father and Son. Because this is one of the early CPRs that was he was trying out in the Antarctic waters. After he left Lowestoft, he went on to work in the southern oceans. And this is Hardy's attempt to address this idea of patchiness. So instead of thinking, goodness, this is just far too complicated, it's just really, you can't get the same data, he set about trying to work out how you could sample patchy environments, something that the ecologist Elton was really sort of amazed at just exactly what Hardy had achieved. Now, the reason for the proud father and child comment was because when Hardy was in the Southern Ocean, when they were trying to put this thing over the side, it was I was going to say it was a side issue. That sounds a bit strange, but you know what I mean. It was something that they were trying out, and the crew kept on calling it the camel's bump. You know, when it had a cover over it, apparently it looked like a camel's bump. One of the crew, not quite so kindly, would call it the horse's bottom, probably the horse's bum, knowing a Lowestoft type approach, but you just don't know. Horse's bottom anyway, HB, and it became a sort of popular thing that it was called HB. When they were down in South Africa, a dinner party off the boat, Hardy was approached by some rather posh ladies who said to him, well, can you tell us why this device is called HB? And Hardy's response was, well, it's rather embarrassing. I've only just got engaged, and they will keep on talking about Hardy's baby. And so in the book that he wrote about those trips to the Southern Ocean, Great Waters, here's the picture of the proud father and child. So when you hear about Hardy's baby, HB, they're talking about the CPR. So here we have when the wanderer, the plankton, meets Hardy's baby. One other part that I want to introduce, though, is a painting that Hardy had when he was at Oxford on his wall. It's a painting which many people describe as being despairing. Certainly Hardy himself talked about this was a time when he was really finding it difficult to see a way ahead. The painting's called Hope. Now, you'll notice that Hope, the character sitting in this globe, is blind, and she's holding a lyre which has only got one string in it. It could be despairing, but one of the things I want to put to you is the attitude that Hardy showed through his life. Even when he was at Oxford as an undergraduate, about to go into the First World War, First World War, pandemic, all sorts of things happen to students, don't they? He had this painting of which was called Hope. Yes, and he was depressive, but notice that even in that case where they're working off Lowestoft, he sees that this is a major issue with patchiness. But rather than giving up, he presses on in hope that he has the hope to get something that works. And his entire career with the CPR, and also when he retired and then headed up a centre for religious experience, the investigation of religious experience, the idea of hope, even against hope for the human race, spiritually, hope for the human race through a lot of the plankton work that he was doing, it is a key feature of Hardy's work and approach. I put together this tiny little riddle. Hardy would have loved this sort of thing, although he would have done it much better than me, and he wouldn't have been critical about it. He would have just smiled and said, well done, Spicer, I'm sure. I am small but a mighty global engine. I wander far but can't move fast. Embroiled in HB twisted mesh I expired, but now live forever in the mind of sages. Breath is my heritage, but you take mine away, you who shot the albatross. what am I? I hope you realise that this riddle isn't just about plankton, but it's about the plankton that have contributed to that CPR and have done far more than just about any other plankton. Not only giving all these ecosystem services, but contributing to our understanding of how the oceans work through the work of the CPR. One of the challenges that I would like to put to not just the next three speakers, but all the speakers who are going to address us is how will what we do with the information that we're going to get during this conference be driven by our hopes and not by our fears. Hope is a central feature of the scientific and personal life of Alistair Hardy. In what way is hope reflected in the life of the CPR? Thank you very much. With that, stop sharing. And next we have an amazing, I would thought, talk by Chris Reid. If there's anyone who can give a history of the CPR, it is Chris. Thanks, Chris.
We're celebrating today the 90th anniversary of the first tow of the Continuous Plankton Recorder across the North Sea on the SS Albatross. The photo on the left shows Alistair Hardy holding the CPR attached to a davit in Hull docks before the ship left for Hamburg. What a fantastic legacy he's left us. More than 7 million miles of the ocean have been sampled since 1931, and more than 250,000 samples of plankton analysed. And now we have 12 other CPR agencies, other agencies that are working in partnership with the CPR programme in Plymouth. A large preserved sample archive exists for other work, and we have more than 2,000 publications published since the survey started. The survey's gone through a number of ups and downs in its history. The first major down was in the Second World War, when everything stopped, starting again in January 1946. In 1989, a number of people were made redundant, and the survey was stopped temporarily over a weekend until a rescue package could be put in place with the help of Bob Dixon, who was then at MAF, Mike Whitfield and Gerald Balch at the MBA, until the Sir Alistair Hardy Foundation for Ocean Science was established to run the survey. This foundation carried on until 2018, and in 2017, a second round of redundancies started that carried on through until the survey was amalgamated into the Marine Biological Association. We've now, against this background, we now, I think, have moved into a new phase of growth. I'm certainly, I hope and expect that this will be the case, and the CPR survey is recognised as a world leader in ocean monitoring. I particularly want to thank the many ships, over 130 of them, that have helped the survey operate over these years. Very many thanks for your assistance, and to the port managers, to the transporters to the ports, and to the ships and crew and captains. Really appreciated. Very many thanks for your help. It's pretty impossible for me to summarize in a few slides the fantastic work that has gone over this period of time. I recommend that people have a look at 2003 paper that summarizes the first 70 years. And again, I've tried to summarize it in a word cloud. You might recognize one or two of the names in there. And in this period, I wish for the years 1956 to 1992 that it should be dedicated to Michael Colebrook, because he played such a fundamental role in that period of time in the CPR survey, and in particular in the computerization of much of the survey work and in research. For the next 20 years, again, I would like to proposed that it be called the Gregory Beaugrand years, because he again played a fundamental role in this interval of time. He published more than 80 papers dealing with the CPR, including a large book. I've got two illustrations from his work, but I just draw attention to the one on the left, which is the showing the very clear northerly movement of warm water plankton to the west of the British Isles, 1,000 kilometers in only a matter of a few years. One of the clear evidences of major changes in the ecosystems around the UK that can be attributed to climate change. I also draw attention to work on fish traits, on copepods and on fish using CPR data. And on the left-hand side, the continuing decline of Calanus vimarcus demonstrated by the survey. With on the bottom left, the alternation that is seen between this cold water species and the warm water species, Calanus elgolandicus. The diagram on the bottom is from a recent paper by Martin Edwards and colleagues that has looked at krill euphorsids in the North Atlantic, and he has seen a 50% reduction in their abundance. And this is one of the few groups of organisms in the plankton that hasn't moved to the north. And so it's seems to be constrained the area that it can now live in. And it's such an important group of organisms for other higher trophic levels. A number of papers have been published on remote sensing over these years. I know in particular work by Dionysius Reitsos, who intercalibrated and validated the phytoplankton color index from the CPR, a visual index of chlorophyll, with fluorometry measurements taken on the PR route between Plymouth and Roscoff. He went on to compare the CPR phytoplankton color index with CWIFS and CZ satellite imagery, allowing him to produce a 50-year plus time series of chlorophyll in the eastern North Atlantic. And in this diagram built on the bottom, you can see the clear jump that took place around about 1987, the 1987 regime shift. The subsequent papers will be, or topics will be covered by other speakers at this meeting, but I wish to draw attention to the huge potential for the use of molecular tools now in CPR archives. Due to initial work by Richard Kirby, who made the breakthrough to show that the polymerized chain reaction could be applied to formalin preserved CPR samples. This work's gone on to look at DNA metabarcoding, a number of other studies, and in particular work by Luigi Vazzulli on a range of organisms, in particular bacteria. And I draw attention to a paper which was published recently on cholera in pathogenic cholera vibrio in Lake Tanganyika at 700 meters above sea level. And I would like people to clap at this moment for Lance Gregory, who went along and brought the CPR and at the same time brought all the samples back for analysis in Italy. He did a fantastic job. And I note at the bottom a recommendation by Luigi that a biodepository of nucleic acid should be produced from CPR archive material. Science policy has been a really important area of work in the CPR ever since it started, and Abby McGuattis-Collop has taken this to new levels, and showing one diagram on the right where she has used data from the CPR with other colleagues from other laboratories at single point sites. I remember early on discussing with Richard Thompson the potential application of the CP archive to looking at plastic pollution, and he's taken that forward in a great way with a recent paper with Claire Osley shown there that shows the huge increase in large particles. And again, this is work that is largely based on the results produced by the workshop of the CPR group. In 2009, I gave a presentation at Ocean Obs in Venice that recommended that a partnership of global CPR programs should be set up to expand the survey to the whole of the world. In 2011, a global alliance of CPR surveys was set up when Peter Burkle was director. And one of the recommendations of this group was that regular global marine ecological status reports should be produced. And some of those that were produced have been fantastic. I draw by Martin Edwards, and I draw attention at the bottom, or perhaps I should note that I was really excited earlier this year to see this paper from Campbell and colleagues, which used the data from all of these surveys to intercompare Calanus as copepod species with temperature to test the Bergmann's rule. And I note the stars on the map showing the locations of the samples, the areas that have been very poorly sampled in the tropics, a future area for the CPR. And I wish to thank in particular Graham Hosey, Sonia Batten, Sani Chiba, and Anthony Richardson for the leadership that they've shown in setting up these other surveys and leading them. The tropics is one potential future frontier for the CPR, but also the Arctic Ocean. The diagram on the top left shows from recent AMIP report, shows the modeled likely positions of sea ice in the Arctic over the next century, with the black line showing the actual observations. And they are going down faster than any of the scenarios produced by the IPCC models, really concerning because of the huge impact that that is going to have on Northern Hemisphere climate. I note that we have been sampling on the edges of the Arctic for many years, but in 2018 started sampling regularly on a route to Svalbard through the Barents Sea. And now we're also sampling in the Bering Sea with the help of icebreakers from Canada, and Jonathan Fitcher will be talking about this. And I note the paper on Neodenticular diatom that came through into the Atlantic Ocean from the Pacific for the first time in 180,000 years, which based on data from the CPR. What a fantastic set of achievements over 90 years that the CPR has made possible. It's been possible really because of the maintenance of standard procedures over the whole of this period of time. That is what the survey is built on over decades, and may it continue for many future decades to provide the material that is proved so valuable up to now. At the same time, we do need to develop further technical approaches to both sampling and to analysis and to investigation and research, but these should not be done at the expense of the core survey. And I draw attention right at the very bottom here to these points, to the need, I think, to provide some support for analysis and technical staff in particular, who have gone through a lot of trauma in recent years, and that also the staff that are no longer with the survey should be given similar support if possible. And I note here also that my great sadness that Gregory Beaugrand, Martin Edwards, and Richard Kirby are no longer associated with the survey. My very last slide is to try and emphasize how important it is that the ocean be sampled, and in particular the ecosystems of the ocean, because of their role in climate change and their impact on living resources. The diagram on the top shows temperature change over from modeled results using the Bergen climate model. And the vertical lines show the boundaries of the plot on the bottom, which are actual observed temperatures from the GISS program in the United States, showing the zonal mean temperature for all the different latitudes. And what really astounded me when I downloaded the data from GISS was to see that the same pattern of Arctic amplification is evident much, much earlier than shown in the model results from 20 years ago. I've been highlighting this in lectures on climate change for many, many times, but people have ignored the fact of the urgency of climate change. Many thanks. Look forward to seeing many of you, hopefully before too long, and best wishes from New Zealand.
I was going to say thanks, Chris, but I mean, it's recorded, so I'll say thank you when you appear in person. If only we could weather our climate change, as well as the CPR survey has weathered political and scientific and funding changes. Next, we've got Alistair, Lindley, Tony John, definitely people who are worth listening to in terms of a personal history behind the CPR. Really looking forward to this.
We can't hear you at the moment, I'm afraid. Can you just check your microphone for us?
Experiences. This will be a short talk. Good morning and welcome to the history of the CPR survey, the people who worked in it and their experiences. This will be a short talk covering the first 2/3 of the survey. The CPR was devised by Alistair Hardy for sampling from the discovery in the Southern Ocean in 1925 to 27. The version he used is now known as Mark I, and was rather unwieldy. According to Hardy's account in Great Waters, it was known to the crew as the horse's bottom. His association with the expedition and the Southern Ocean was commemorated by postage stamps which feature the CPR Mark II. This was developed when he was professor at Hull for sampling from ships of opportunity, and we commemorate the first tow on the 15th of September 1931 today. During this tow, Hardy broke his finger, saving the CPR from being lost. The start of the survey. This was at the University College of Hull, and other scientists involved at this time were Cyril Lucas, second from right, and George Henderson, who was third from right and shaking Sir Alistair Hardy's hand. Alistair Hardy was appointed professor at Aberdeen in 1942, and he became Linacre Professor at Oxford University in 1946. He was knighted in 1957. He wrote 2 books in the Collins New Naturalist series, The Open Sea, The World of Plankton, and The Open Sea, Fish and Fisheries. He also wrote the one about his voyage to the Southern Ocean called Great Waters. Cyril Lucas was officer in charge at Leith from 1937 and became head of department in 1942 when Hardy moved on. He became director at Aberdeen Lab in 1948. He was awarded the CMG, Call Me God, in 1956 and knighted in 1976. George Henderson stayed with the survey until his retirement in 1969. Regular sampling started in 1932 on 3 routes from Hull, shown here in black, with the extensions later in the 1930s, shown in red. An outstation was opened at Leith in 1937 to service the more northerly routes. The staff increased, including Peter Ray, who succeeded Cyril Lucas as head of department when Lucas moved on and headed the survey until 1957, when he was appointed Professor of Biological Oceanography in Agricultural and Mechanical College of Texas, and subsequently went on to the University of Alaska. Colin Rees, from 1937 to 56, was very productive in the post-war years, including much work on Marrow Plankton. However, the use of concentrated HCl, which reacts with formalin, to clean bivalve larval shells may have contributed to his premature death in 1956. Most analysts seem to be relatively long-lived, despite routine exposure to formalin and tobacco smoke, which was normal before health and safety regulations were enforced after the move to Plymouth. In a 1937 paper in Oceanographical Research at the University College of Hull, 1931 to 36, talking about the CPR, Alistair Hardy said, "We aim at linking up the variations in the plankton distribution with climate changes on the one hand and the fluctuations in the fisheries on the other. This shows how far ahead of his time he was. From September 1939, there were impediments to navigation due to World War II, with many members of the team serving in the armed forces, until the survey resumed in 1946. Roland Glover, shown here top left, was appointed to the department in 1945, and he became officer in charge of the Edinburgh laboratory on Peter Ray's departure. He became director of the lab in 1967, and when IMER was formed, he became director and moved to Plymouth. He retired in 1983. Sampling was re-established in the North Sea and the V route to Iceland in 1946, together with tows to ocean weather stations in the eastern North Atlantic. The British Ocean Weather Ship base was in Greenock, near Glasgow. CPRs were delivered in laboratory vans from Edinburgh, and analysts were occasionally taken as a special treat. Among the staff appointed in the early post-war years were Dora John, shown bottom left, who moved to Plymouth with the survey, Gordon Cooper, who retired when the Edinburgh lab closed, on the right-hand side of the other photo, and William Brown. William first worked on early development of the UOR and developed the BrainCon recording thermograph in the mid-1960s. First, this was the first recording of physical variables in the CPR. He moved to Plymouth in the early days of IMER and worked on other programs. Dora is shown preparing a figure for photographing for publication in the first days, long before computerized image preparation. Gordon and the other person, possibly Colin Rees, were notably without lab coats or any form of protection from or extraction of formalin fumes. In 1950, the department at Hull was closed and the survey was concentrated in Edinburgh under the Scottish Marine Biological Association, soon being established at 78 Craighall Road, known as the Oceanographic Laboratory. Jerry Robinson, shown on the left, was appointed to the survey in 1951, specialising in phytoplankton. He became head of the survey and was appointed assistant director of IMER. He continued as head of the survey until his retirement in 1986. Michael Colebrook, shown on the right-hand side, was appointed in 1956. He introduced statistical rigour, including changing phytoplankton methods to make the data compatible with statistical methods, and eventually introducing computer use. He was the scientific face of the survey at conferences for decades, and Colebrook and Robinson were the co-authors of many papers. Michael was head of the survey during a critical period from 1989 to 1991, leading to the start of SAPPLES. From 1957, under the direction of Roe Glover, the survey was extended to the Western Atlantic, initially on the Z route from Iceland, with other routes further south and including the routes to the Western Atlantic weather stations. This was supported by a contract with the United States Office of Naval Research. There was an increase in staff, of whom Harry Hunt deserves a particular mention. He was appointed in 1960, working with Michael Colebrook on developing data processing methods and computerization, as well as being a full-time analyst. He was for a while senior analyst and data manager for the survey into the early days of SAPPLES. Another long-serving analyst was John Roskell, starting in 1962 and staying with the survey until his retirement in the early 1990s. On the retirement of George Henderson, ex-merchant Navy captains were appointed to maintain relationships with the ships due to increasing complexity and frequency of change in shipping patterns. John Beatson from 1969 to 1991 and Peter Pritchard thereafter. Without the willing cooperation of the shipping companies, ships, masters and crews, the survey could not exist. We owe them a great debt of gratitude. Alistair and I were appointed respectively in 1967, me. I overlapped with George Henderson, the only remaining member of the 1931 staff, and Alistair in 1969. Computing became important in the 1960s, but even when we started, cards were punched at the laboratory and sent to the regional computing centre, with the output on fan-fold paper returned the following day. Routine data processing was still manual, and each of us had to spend about a third of our time on data processing. Over the next few years, computerization took over, but some of the characteristics of CPR data processing dated from the limitations of punched cards. And now I'll hand over for the second part of the talk to Alistair.
A notable appointee in 1967 was Bob Williams, who you see third from the left on this picture, who initiated and ran a programme of sampling from ocean weather ships with the Longhurst Hardy Plankton Recorder in 1971 to 75, withdrawn. This mainly provided information on the vertical distribution sampling of the CPR. In late 1969, the intention to create IMA and the move to Plymouth was announced. The following year, the Rothschild report introduced the customer contractor principle for scientific research funding. This led to the contract from MAF, which later DEFRA, which was critical to preserving the survey. New members of staff in 1970 included Chris Hopkins, whose red hair you see on the left of the photo, who became professor at the University of Tromsø before becoming Secretary General of ICES from 1994 to 1999. Chris Reid joins the CPR team in 1972, and we will mention him again. The move to Plymouth was piecemeal. We moved in late 1975 to 76 into temporary accommodation in now demolished buildings on Mill Bay docks. Through the long dry summer of 1976, this was not bad. However, when the rain started, the buildings leaked like a sieve. New staff were beginning to be recruited to the team, notably Tanya Budd, later Tanya Jonas, who eventually became senior analyst. The IMA building, now PML, was opened in 1977, and the Edinburgh lab was closed with the departure of many staff, so further staff were recruited in Plymouth. The Edinburgh lab was gutted, and all the furniture, fixtures and fittings were sent to Plymouth. Much of this was not needed, and staff were allowed to help themselves to items they could use. The new building had been designed with features to accommodate the survey, which had been intended to be using UORs by this time. These would have been delivered through the gate on the extreme right of the picture to the workshop, which never performed that function and was completed as the seawater hall. Health and safety regulation, together with the need to exclude formalin fumes from parts of the building where experimental work was in progress, led to the development of extraction systems so that analysts were no longer inhaling the fumes. By 1976, the end of the US Office of Naval Research contract and other budget limitations had led to sampling being suspended west of 20 degrees west. So the diagram on the left shows most of the routes in use at the time. Jerry Robinson retired in 1986 and Bob Williams took over as head of the Oceanic Ecology Group. In 1988, PML was formed from IMA and part of the MBA. At this time, NERC was moving to the direction of prioritising funding projects with defined outputs within 3 to 5 years, not open-ended programmes, and the CPR survey was declared a unit of redundancy. Some analysts were redeployed to other PML programmes, in some cases returning to the survey later. It was almost immediately the significance of climate change and the need for long-term data sets were recognised, and eventually NERC became a substantial supporter of the survey. The survey was salvaged by the ongoing MAF contract and the contract from the Department of the Environment, now part of DEFRA, to which Chris Reid was seconded at the time. From April 1979, a reduced team of 5 analysts continued, financed by the MAF and DOE contracts, and with Michael Colebrook in charge. SAFOS was formed with noticeable support from Bob Dixon, right-hand photo of MAF in the late 1990s, in late 1990, and was functioning by spring 1991. With renewed funding, the survey expanded back into the Western Atlantic. Michael Colebrook retired in 1992, and John Gamble was appointed director. The survey was relocated to the MBA at Citadel Hill in 1993, and other new staff were recruited. After John Gamble's untimely death in 1994, Chris Reid was appointed director. Under his dynamic efforts, more funding was raised, and the survey expanded to include routes in the Mediterranean and the Gulf of Guinea for a few years. And the first pilot tow in the Pacific in 1997 was followed by regular sampling from 2000 onwards. The CPR was deployed by other institutions in other parts of the world. The staff numbers increased, including Martin Edwards, who eventually became deputy to several directors, Sonia Batten, who went on to Canada to run the Pacific CPR routes, and David Johns and Marianne Wootton, who remain mainstays of the survey. Later arrivals, such as Gregory Beaugrand and Anthony Richardson, were responsible for some of the most highly cited papers resulting from the CPR survey and the recognition of the importance of its data in studies of the impact of climate change. Claudia Castellani was the driving force promoting the production of the book Marine Plankton, to which many members of the CPR team contributed. Richard Kirby developed a method for analysing DNA from the CPR samples, enhancing further the value of the historical sample. Directors following Chris Reid from 2007 were Peter Burkles 2011, Nick Owens to 2015, and Willie Wilson thereafter. Knock-on effects of the financial crisis of 2007 to 2008 led through to reductions in public funding, and once again financial problems mounted, resulting in the reductions in the survey, painful redundancies, and the merger of the SAFOS with the MBA. We have mentioned only a few people that are involved in the survey, concentrating on scientific staff of the analysis team, but we apologise for many omissions from this talk. We should also acknowledge the work of the workshop and admin staff, without whom none of this would have happened.
Wasn't that absolutely incredible? Goodness. Yeah, I mean, what do you say? Just a history of people as well as a history of science, and both equally important, both equally impressive. We're going to finish this session with an introduction to the plankton. Marianne Wootton is going to do what Sir Alistair Hardy did in the 1950s, going to introduce us to the beauties of plankton, taxonomy, and why we care.
Hello, my name is Marianne Wootton, and I'm Senior Plankton Analyst for the CPR Survey here at the MBA in Plymouth. And I'd like to talk to you today about what is plankton, what is taxonomy, and why should we care? Hopefully this will be an introduction to the flora and fauna of planktonic life in our oceans. And hopefully we'll look at the hidden beauty of the microscopic world and the huge variety of life found within the plankton, and talk taxonomy, an tedious or a powerful tool to catalogue and capture biodiversity. Well, what is plankton? Imagine, if you will, lying on your back in the sea, letting the ocean carry you. This is what plankton do. The word plankton comes from the Greek word planktos, meaning to wander or to drift. And aptly named, these tiny plants and animals are at the mercy of the elements that support them. So what type of organisms then do we find in the plankton? Well, let's take a look. Let's start with the phytoplankton, the plant plankton. These are typically algae. We have examples, and these guys will possess pigments such as chlorophyll. They will take in carbon dioxide from the water, sunlight, and they'll use those things to produce sugars and then pump out oxygen. We also have the zooplankton, or animal plankton. zooplankton will feed on the phytoplankton. And within the zooplankton, we have a whole range of organisms. We have some single-celled zooplankton and some multicellular life too. Worm, here we have mollusks. So zooplankton will feed on the phytoplankton, the planktonic, and of course zooplankton themselves will be food for other organisms such as larval fish. So plankton actually, it encompasses quite a big size range. So bacteria and viruses are part of the plankton. And then everything in between, the size range goes all the way.
Hello, we just seem to be having a bit of an issue here with the audio. Just bear with us for 2 seconds, please. This is the nature of virtual events.
So plankton, they will take in carbon dioxide from the water, they'll use sunlight, and they'll use those things to produce sugars and then pump out oxygen. We also have the zooplankton or animal plankton. These zooplankton will feed on the phytoplankton. Within the zooplankton, we have a whole range of organisms. We have some single-celled zooplankton and some multicellular life too. Here we have a type of polychaete worm, here we have a type of mollusk, and here we have a small crustacean. So zooplankton will feed on the phytoplankton, the plant plankton, and of course zooplankton themselves will be food for other organisms such as larval fish. So plankton actually, it encompasses quite a big size range. So bacteria and viruses are part of the plankton, and then everything in between, the size range goes all the way up to jellyfish. Yes, jellyfish are part of the plankton too. And plankton are found from the very surface of the ocean all the way down to the deep ocean. And this means then that plankton forms a huge part of life on Earth. They're single cellular, they're multicellular, and they're found on a vast part of the planet. Well, why do we study plankton? Well, very briefly, phytoplankton produce around half of the world's oxygen, so every other breath is a plankton breath. Phytoplankton are known to produce a chemical called dimethyl sulfide, which we know is involved in some cloud formation. Plankton are deeply involved in the biological pump, drawing down carbon down to the deep ocean, and thus drawing carbon dioxide out of the atmosphere. So plankton are hugely important for the carbon cycle. Of course, phytoplankton and zooplankton form the very base of the marine food web. So no plankton, there would be no fish, no crabs, no starfish, and of course no charismatic megafauna, such as whales and dolphins, seals and turtles. Plankton are very sensitive to their environmental conditions around them, such as temperature, nutrients, salinity, and thus these make ideal organisms to study changes in the ocean. And some plankton, some of the algae, have human health impacts too. They can form harmful algal blooms, which produce toxins, which if they're ingested by us or other animals, well, they can make us quite poorly. So what organisms do we find in the plankton? Here we have a diagram of the tree of life. This represents all life found on the planet. So hopefully you'll agree with me that there's a huge range of life that we find in the plankton. Almost all of the tree of life we find in the plankton. And I've just got a few moments to try to put across the beauty and the diversity of plankton. It's a tall task, isn't it? But let's have a go. So what organisms do we find in the plankton? We have the plant plankton, of course, the algae. And here we have an illustration by Sir Alistair Hardy. Sir Alistair Hardy, of course, the inventor of the CPR. He was a scientist, but he was also a very accomplished artist, and he would draw what he saw down the microscope. We have a range of organisms here, mostly they're diatoms, these big cells here. We can see some form chains, we can see some are long and thin. We have a chain-forming species here. We also have some dinoflagellates here. These are single-celled organisms that photosynthesize. These have a flagellum that goes around their middle, their girdle band. I'd like to talk perhaps a little bit more about the diatoms, an incredibly common group found in the plankton. What other organisms do we find in the plankton? Well, we don't necessarily always need a microscope to see some of the phytoplankton, to see some plankton. Here we have a man who's walked along a beach, and where he's walked we can see these blue flashes. And these blue flashes have been caused by this organism here. It's a dinoflagellate known as Noctiluca scintillans, which translates as night sparkle. What a beautiful name. So these single-celled organisms are able to bioluminesce. They contain an enzyme called luciferase, Lucifer as in the bringer of light, and when they're disturbed they bioluminesce. And we can see this with our naked eye, at least when it's dark anyway. On the left we have a picture, a satellite picture of the UK, the southwest of the UK. Plymouth is somewhere around here. And here we have an organism called a Coccolithophore. This is known as Emiliana huxleyi. It's about 1/100th of a millimetre in diameter. And this organism can form huge blooms. And this milky blue area that you can see on the map down here has been caused by millions and millions of these organisms. So huge blooms of plankton can be seen from space. Well, what other organisms can we find in the plankton? We find plants, we find animals, and also we find things in between. They're known as mixotropes. Here we have an organism, a single-celled organism known as Dinophysis. It contains chlorophyll, it can photosynthesize, but also it will actively prey on other smaller types of animal plankton. So imagine that. Imagine if you yourself, perhaps you've missed your breakfast, you're a bit hungry, you haven't got time to go out and go shopping and get a snack. You could just sit outside, get a bit of sunshine, top yourself up, and then get ready for the rest of your day. How amazing is that? What an amazing adaptation, mixotropes. Here we have another dinoflagellate. These are known as warnoids. And this time these guys have this structure here. Let me just highlight that for you. They have an eye. They have an ocellus. They have some kind of rudimentary retina. They have an eye lens with pigment. And this one on the right here, we can see that it's engulfed, it's ingested a copepod, an invertebrate egg. So imagine that, a single-celled organism that's managed to develop its own eye. And we know that eyes throughout evolution have evolved independently of each other, and that's what's known as convergent evolution. But who would have thought that we would see eyes in a single-celled organism? I just find that amazing, and we see these guys in the plankton. No talk about plankton to be complete without talking about a guy called Ernst Haeckel. He was a naturalist in the 1900s, a scientist and a very accomplished artist, and he drew lots of types of plankton. I'm very quickly running through some here. These are copepods, my favourite type of plankton, very beautiful creatures. And we have some single-celled organisms here on the right, known as radiolarians. And here we have another example of an artist who is inspired by plankton and by Haeckel's work. We can see this image of a jellyfish drawn by Haeckel on the right. And we have this artist and architect known by the name of Hendrik Bligh, known as the father of modern architecture in the Netherlands. And here we can see he's drawn this image of an Art Nouveau electric lamp. Also, Hendrik, still inspired by plankton and Haeckel's work, he drew these lovely pictures of jewellery and chains. And for those of you who know your plankton, you might recognise the links in this chain, this necklace, as the cell odontella. So how wonderful is that? The beauty of plankton is captured in some of the art around us. Let's look at the animal plankton, or the zooplankton, and we can split the animal plankton into two groups, one of which is the holoplankton. These guys will spend their entire life as part of the plankton, and here we have some examples. This is a krill, or a euphausid, and we know that these guys form a very important part of the diet of whales. Of course, they eat them by the ton. Here we can see some eyes at this end. These little red dots, this pigment indicates the presence of photophores, and we know that photophores are cells which emit light, they bioluminesce. Here we have another small crustacean known as a copepod. Copepods are an incredibly important food source for things like larval fish, and copepods are a very diverse group. There are over 2,000 species of marine planktonic copepod. Some other interesting organisms here, these are pelagic mollusks. This is known as a sea angel, and this is known as a sea butterfly. They're given very romantic common names, aren't they? But in real life, well, perhaps they don't quite deserve it, because this guy will feed very voraciously on this guy here. Then we have the other part of the plankton known as the Marrow Plankton. And these guys will only spend part of their life up in the plankton, and then typically they will settle down and form part of the benthic environment, perhaps as crabs or barnacles, that type of thing. So the Marrow Plankton are usually larvae of other animals. And here we have a crab larvae or a decapod larvae. We can see its eye here. We can see these spines on its dorsal or its back, and this big spine on its front, on its rostrum here. These spines just aid in flotation, and I guess they're also a deterrent to predators. You wouldn't want those nasty prickles in your mouth, would you? But interestingly, let's look at the form of this guy. And it's something we see a lot in the plankton, is that the larvae don't look anything like their adult form. And if we think of the terrestrial environment on land, well, that's similar to tadpoles and frogs, isn't it? So we know that tadpoles undergo some kind of metamorphosis, and eventually they turn into frogs or toads. Well, we see that same kind of pattern in the plankton too. And here's just another example. Here's an echinoderm or starfish larvae. And again, we can see that its larvae, well, it looks nothing like its adult form, does it? What is taxonomy? Well, in its loosest, all-encompassing sense, taxonomy is just purely describing organisms. And if we think about ourselves, how would we describe ourselves? How would we do taxonomy of the human? Well, we'd perhaps start with a drawing or a photograph, and then we'd add some description. We're a vertebrate, we've got 2 arms, 2 legs, fingers and toes, 2 eyes. We'd describe the environment that the organism was found in. So in the case of us, we're found on land, we breathe air. And then we'd try to describe, well, who are the other family members? Who looks like us and who doesn't look like us? How are we similar and how are we different? And then once we'd done that, we'd attempt to kind of put ourselves in some kind of evolutionary context. This is essentially what taxonomy is. And we use this hierarchical system here. This system was pretty much invented by a guy called Carl Linnaeus in the 1700s, and this system is still used today. It's a system of essentially cataloguing all organisms, and we use this hierarchical terminology. So again, let's think about ourselves. We know that we're animals, we belong in the kingdom Animalia, we're part of the phylum Chordata, we have a backbone, we have a spinal cord, we're mammals, we're part of the primates, and then very much down the bottom here, we're known as Homo Sapiens. So each species on Earth will have 2 names associated with it. It's what's known as binomial nomenclature, 2 names. And of course, we're known as Homo Sapiens. And this method of cataloguing organisms really does help us. If you think there are millions of organisms on the planet, how do we keep track of them? Well, we very much sort organisms into these different kingdoms, phylums, class, orders. That's how we keep track, really. And the further down the hierarchy you go, that's just the finer detail. So if you imagine perhaps that all life on Earth is catalogued in some library somewhere, well, this system would tell you perhaps on what floor in the library to go to find a particular species, on what shelf you would find a species, in what book you would find that species, on what page and on what line. But taxonomy has had, well, a bit of an identity crisis, I guess, in the last 30 or so years. It's not seen as a particularly sexy science, let's face it. And it's definitely difficult to get recognition and funding to do taxonomy. So imagine, if you will, all of the books, all of the programs you've watched about animals, about organisms, when you read about an ecosystem and you read about all the biodiversity there, how often is it that you see in the references the person that first described the organisms that you've been reading about? How often is it that you see the reference for perhaps the identification books that were used, so as that the scientists could identify their animal they're talking about in their ecosystem paper or their TV program that you're watching? It just doesn't happen very often, does it? And so, because of that, it's difficult to get funding. And, okay, taxonomy, it can be a bit tricky, can't it? So there's generally name changes. Yes, names keep changing all the while, and with advancements in technology, with molecular work, when we start to look at DNA, we can find species where we didn't think there were species before, perhaps. But taxonomy really is just a filing system that we use every day. And if I were to present you with this group of animals here and ask you to separate these animals into two logical groups, I imagine what you'd do is you'd split them up into cats and to dogs, into the felines and into canines. And I'm pretty sure you would find that quite easy to do. But what you've done there is a bit of taxonomy. You've identified your organism and you split them into similar looking types and you've labelled them differently. That's essentially basic taxonomy. So we do it every day. It doesn't have to be hard. So why is taxonomy important? Well, let's perhaps flip that question and say, why is taxonomy important to us? We're a very egocentric species, aren't we? We like to know what things affect us. Well, for us to be successful, we need food, we need water, we need clean air, we need shelter, we need medicine, and perhaps, maybe when there's spare time, something nice to do and something nice to look at. And so that means we need our planet, we need the life on Earth, our ecosystems to be healthy, don't we? And those ecosystems, it could be big, it could be the whole planet, or it could be a small ecosystem like perhaps one river. But we need to know what's happening in that ecosystem, and whether it's healthy or not. And a part of measuring an ecosystem's health is to look at its biodiversity, isn't it? But biodiversity is taxonomy. Biodiversity describes the number of animals in an ecosystem. But how do we get to that? Well, by using taxonomy, of course. So all of those millions of creatures we find on the planet, they're all deeply involved in taxonomy. So without taxonomy, we can't understand biodiversity. And thus, without taxonomy, we just don't understand life on Earth. Thank you very much. Hopefully I've given you a small introduction into the world of plankton, its beauty, into taxonomy, and why taxonomy is an essential science. Thank you.
That was great, Marianne. A really simple but actually quite detailed and wide-ranging introduction to plankton and why taxonomists should be paid. So we're now moving over to our discussion time, and Maya, you're going to help by giving the questions, I understand?
Yeah, lovely. Thank you, John. Can I just ask all of the speakers from that first theme to please switch your cameras and your microphones on so we can all see your lovely faces? That would be fab. We have got a couple of questions. And again, if you wanted to ask a question and say it live, then please do. You can do that by clicking the raise hand button, and then we can activate your microphones. So feel free to do that. Always nice to have some interaction. Lots of lovely comments from people saying what fantastic talks, and great to have such an amazing overview of all of the great and good that have made CPR what it is. So thank you to all the speakers. So one of the first questions that we've got here is from Michael Poulston, who's saying, Chris, what are your thoughts on industrial krill fishing in the southern oceans and Antarctica? There seems to be increasing evidence that the fisheries at such scale is negatively affecting the wider ecosystem. Chris, might you be able to help with that one?
I will do my best.
Thank you.
I think you can make the analogy with the seas around the British Isles and the North Sea, which are overfished, and we've hardly got any large fish left, and there would have been in the past. And it's the larger fish that produce more eggs. We've overfished that part of the world, and I think we're possibly doing the same in the Southern Ocean, and certainly we have with the whales. The whales are coming back, but there is no way that they are anywhere near the numbers that we've had in the past.
Great, thank you, Chris. And does anybody else have anything they'd like to add to that? No, okay, great, thank you, great question. And we've got another one about modern technology. So obviously we've got lots of new bits of kit out there. This is a question from Facebook that allow us to be able to identify species through imagery. So how does this compare to our extremely experienced analysts who can identify it by tiny little details? So do you think there's a sort of a possibility that this technology might advance and exceed the skills of our analysts?
It would be good to hear Marianne, Alistair and Tony on that one.
Hi, can everyone hear me?
Yes, we can.
Yes, thank you. Yeah, I might just jump in there or at least to begin with. It's a really good question, and it's something that lots of people in the plankton world are looking at. It's this kind of automated AI form of analysis. Superficially, it looks like that type of analysis, the computer generated stuff, would be a lot quicker than traditional microscopy. But I guess one of the success stories of the CPR is that we identify taxa to species level. And that's really important because different species will perhaps inhabit different thermal niches. So different types of copepods perhaps might like cold water, or some might like warm water. And you would only tell the differences between those different types of copepod by looking at the really fine detail. And I mean flipping the animal over, perhaps feeding through its legs, getting in a needle and kind of digging in through the legs and looking at maybe, you know, a small part of its body and looking at the shape of the curve on maybe one part of its leg. And that feature that you're looking at to discriminate between species might only be 1/100th of a millimeter in size, you know. So I think modern technology just isn't up to that kind of standard yet. It can't flip animals over. Let's imagine that you've got image analysis and you have your animal lying on your glass plate or whatever. It might be lying in the wrong orientation for you to tell what species it is. It might have legs overlaying the bit that you need to see. So until we develop the technology to flip the animal over and flick through its legs, well then I think we've got some way to go. But having said that, of course, technology can identify things to broad groups. So it could be that this AI type technology could identify something as a copepod or a decapod or a worm, which of course has value too. But to species level, I don't think we're quite there yet.
I think I'd just like to say that ever since I started being an analyst, we've been told that within a few years, computer image analysis would replace the microscopy. It hasn't happened yet, but technology advances, and we'll wait and see.
I would agree very, very much with that. The importance of identifying individual species cannot be overestimated.
Great. So it sounds as if our analysts are still going to be in work, which is fantastic. Lots of very skilled people here within the MBA and that work on the CPR survey over the years. So lots of inherited knowledge there. And we have got a hand up in the audience. So Paul Hart, if I just switch your microphone on, and then if you'd like to ask your question to the panel. Jack, that might be down to you to switch the microphone on. Just dealing with the technology 1 second, and then we should be able to hear you, Paul.
Is that it? Can you hear me?
Yes, we can. Thank you, Paul.
Tony and Alistair, your talk was very interesting. Obviously, from someone who's been involved, it was interesting to hear the story. I think one person who you left out, who I think had very significant influence on the data analysis, was Mark Williamson, who was employed at the CPR in Edinburgh for a fairly short space of time. But I think I'm right in remembering that he was the one who introduced principal components analysis into the analysis of the data. And then, of course, Michael then took that up and ran with it and developed it much further. But I think Mark was someone who set the process going. Am I right or am I wrong?
I think you're quite right, but I think we excluded him from the talk because he wasn't actually a CPR analyst. He was employed at the Edinburgh laboratory. It was on plankton indicator samples. And of course, the plankton indicator was another of Hardy's developments, but he wasn't actually directly involved in the CPR survey. But you're quite right, it was he who introduced, well, probably the first to use computing and the first to use the principal components analysis.
I think just as an aside and completely irrelevant really, but Tony, you must be one of the greatest survivors, having survived years of working with Dora John in her office, who smoked like a chimney, and also breathing in formalin over the years from an open stage.
Yes, it was it was very different in those days. The fact that we breathed in some cases very very concentrated formalin. I mean, I do remember cutting some roots where we were working with 40% formalin and no formalin extraction, which seems almost impossible to believe nowadays. But in fairness to Dora John, admittedly, she did chain smoke in the same laboratory as we were analysing samples, but she had the goodness to have the window open.
Well, I'm pleased to say that the practices today are a lot tighter, and our team are well looked after. We have got another question. Thank you, Paul, for that. We've got another question here asking about funding for taxonomy and how best to improve that.
Okay, I guess that's for me. Funding for taxonomy, wow. That's a very good question. I think taxonomists are kind of, we hide our light under our bushel, I think, a little, and don't quite state the importance of taxonomy enough to the right people. So I think it's kind of having those kind of wider conversations. Like I said in my talk, you know, if we want to understand biodiversity, well, we need taxonomy. You can't do one without the other. You need good taxonomists to inform biodiversity studies. So I think it's including taxonomists in those kind of works and giving them reference. Yes. Yeah, when as I say, when someone writes a paper and they give you a long list of organisms, well, you should have somewhere referenced the person who first identified that organism, surely.
Yes.
Has anyone else got any ideas? Yeah.
Yeah, I think I would like to say that yes, taxonomy is the foundation of all biological work, really. But the trouble is that the taxonomy is not highly cited. You get the few papers which are directly on taxonomy, which might cite taxonomic work, but once a species is categorized, then you don't tend to list the authority in the bibliography. So citation indices underrate very highly, I think, the importance of taxonomy.
I do remember in the 1990s, the government realised that they were not just running out of taxonomists, but their work was suffering because they hadn't actually employed people, particularly to academic posts, because it was said taxonomy was mere natural history, as if that's something bad, but they wanted proper scientists. And a lot of the research in universities was grinding to a halt because there weren't enough taxonomists. And one of my PhD supervisors, Jeff Moore, I remember in my PhD, I'm an ecophysiologist, him saying to me, you should read Paul Ehrlich's book on A World of Wounds, where he recommends that every biologist should sort of give back 1/10 of their time to something in biology that no one's going to pay for. And he said to me, you know, okay, you're an ecophysiologist, but make sure you do some taxonomy of the groups that you're working in as well. It's an interesting idea. I don't know how many people ever took Ehrlich's 1/10 of their time up, but it'd be a shame if taxonomy was only 1/10 of the time of practicing scientists.
Great, thank you. And that sort of answers the next question, John, actually, which was about whether there's enough focus on taxonomy in university courses to provide the workforce for the future. And if there's anything that we can done at the universities to improve that situation, which you've sort of given a sort of potential solution for. Does anybody have any other comments on how it could be improved within universities? Yeah. Okie doke. And just a couple of comments. Oh, Willie has got a question. Hold on, let me just unmute you there, Willie. Yes, you should be able to speak now.
Yeah, sorry. Yes, it was just a comment on that. I mean, I think it is pretty sad that universities don't teach taxonomy like this. And I've been involved in quite a few taxonomy workshops over the years, and one of the things I find frightening is the age of the teachers at these, that it's the same teachers that have been doing it for about 50 years, and there really isn't a cohort of new academics coming up through the system. I mean, the CPR survey and it's all, you know, the sister surveys in Australia and over at the Pacific as well, and Rana in Cyprus. There's a sort of a few examples of it, but there's just at the university level, it just doesn't seem to be done at all. And I'm not quite sure why. I mean, maybe John, do you have a comment on that? I mean, I guess it's
Yeah.
I mean, there's a lot of I mean, even in some of the classic departments like Cambridge or Oxford or Reading or you know, Birmingham, some of these places. I mean, are they even teaching taxonomy? Maybe at some of the higher levels, but I'm just not aware of anybody that does it.
That, I mean, we've always had this problem with teaching taxonomy. There just aren't the people to do it, and then it gets to a critical level, and the government says, "Oh, we need to invest in this." And I do remember, I think it was the late 1990s, the government actually put a lot of money into putting taxonomists into universities. But it was a sort of one-off panic because things got really bad. And I suspect it'll be the same again. I mean, I teach biology and marine organisms, and I'm probably the most qualified in terms of knowledge of systematics and taxonomy to do that within the university, which is quite sad, really, because I'm an ecophysiologist, you know. But yeah, it doesn't seem to attract quite as many people. I'm saying that, but there are some students who come through and they just get really excited about what taxonomy is for, and will learn taxonomy, but then getting jobs in that. Well, I think we have to wait till there's another crisis and we can't actually identify our biodiversity before the government does things.
Yeah, and I think that, I mean, there's a lot of school of thought that thinks that molecular biology will replace sort of classical taxonomy, or even, I mean, there's comments being made around some of the work that Pierre's going to be talking about, the particle imaging. But I mean, my argument every single time is that you can't verify any of these techniques unless you under the lens of a proper taxonomist, really. And there is a severe lack of taxonomists coming up through the system.
Yeah.
And I think we need to be jumping up and down to sort of try and help with this. With molecular biology, I am a molecular biologist, and you know, I'm one of the biggest advocates for DNA-based technologies, but it's only a starting point. You need to have that taxonomic, the physiological expertise to actually make sense of what the DNA is actually telling you. So, but the other point I was going to make, particularly around some of the imaging techniques, one of the ways I often explain it is I'm a big fan of these and also molecular biology, but it should be layered. You know, so if you sort of layer it on top of a core of taxonomy data, and you can sort of start increasing the granularity of the type of data that you're getting, and you can make, you know, there are strengths to some of these techniques, and Maz said that sort of quite eloquently. There are other things that you can get from these other techniques, but you'll never replace taxonomists. I got into trouble by the CPR taxonomists by putting a slide up once of an imaging system and flow cam system and said, "Meet our new taxonomist," and I got quite rightly hammered for it. So.
I mean, you say we'll never replace taxonomists, but I don't think we'll have many taxonomists to replace.
Yeah.
And teaching very large classes, not just at first year level, but even in honours level, where you've got 50 and 60 in a class, trying to teach taxonomy properly, it's quite a challenge. We need different ways of thinking about this, which involve the bioimaging, which involve the molecular, and involve that ability to look and to see. I mean, looking and seeing is the hardest skill for any biologist.
Interesting discussion, which seems to be causing a little flurry of hands to go up here. So we've got Dr. Tom Doyle and Paul Hart and Gerald Bolt. So just wondering if any of you are wanting to respond directly to that discussion on taxonomy.
Yeah, I was just going to come in on that because I teach invertebrate zoology, second year students here in University College Cork in Ireland. And I guess the thing about it is that class sizes, I think somebody's just mentioned it there on the chat, the class sizes have increased, and it's become increasingly difficult to give students enough time using stereoscopes and identifying particular organisms. So that's kind of been one of the biggest challenges for us is actually class sizes have become so big, so practical class time has been reduced, and various different things through the different years fall off, and taxonomy has been one of those. In favor of maybe more molecular practical classes. But to kind of get around that, like we've just set up what we call a Plankton Club here in the university, where it's totally voluntary for students to participate. And we've had a really good uptake in that. So students can come in and they get to spend time identifying zooplankton, and the feedback has been fantastic. The students just love it, that, you know, to come in and have this freedom to look at zooplankton and try and improve their taxonomic skills. Many of them are not particularly interested in plankton when they come along, but after a bit of time, they just love it.
Love the idea of a Plankton Club. I think we should have a twin club over here in the MBA as well. Great stuff. Thank you, Tom. And I've got a hand up from Gerald here. Gerald, would you like to ask your question? You should be able to speak now, Gerald.
Yes, can you hear me?
Yes, I can. Hello, Gerald. Welcome to today.
I should just point out that I was the last phytoplankton taxonomist to work at the MBA.
Great stuff. Thank you, Gerald.
Nobody was trained to follow me.
Gerald, why was that?
I think people are just not taught taxonomy at university, and therefore they don't think it's a valid thing to work on. But I was trained as a plant taxonomist at university.
Well, hopefully
chip in there with a point. Can you hear me?
Yeah, we can. Yeah.
No, I just think that from a university perspective, I think now that courses are so student orientated, so the idea is that you can't offer courses which students might find boring. And I think taxonomy has become regarded as being something which is a bit dry, not very interesting, and it's overtaken by molecular biology and behavior and things like that. And also, I think many students come to university with very little knowledge of natural history. I always remember saying to a group of students, you know what a swallow is? And there were just blank faces all around. Not a single student knew what a swallow was. And that just amazed me. And that was some years ago. So I think that it is a difficult topic because we are now so driven by the customer-orientated education process, which thinks that you've got to offer what you think students are going to like, not what they really need.
Okay. Should I say that a few years ago, when the Liverpool class was at the MBA, I gave a talk to them on algal taxonomy. And I gather that one of the students said, we never realised that the algae were so interesting.
And that's what we need, these engaging, inspiring individuals to get the next generation to want to study that topic. I've just got another question that I'd like to ask. There's a question here, slightly different, but also looking at the sort of the future. Do you think that in future you could genetically identify each ½ inch of CPR silk by presence or absence only? I'm not sure if that's one for Maz, perhaps? Or David even. David, if you wanted to switch your camera on, if you're there.
I could jump in and just say theoretically, yes.
I was going to say, ask Willie, ask him about eDNA.
There we go.
You could, I mean, actually, very easily you could do it, actually. But it's something we've been talking about as well.
Great stuff. Well,
it wouldn't tell you everything. And somebody, you could argue, might not tell you very much either, but it would give you a lot more information about a lot of organisms that you might not know very much about as well. But of course, that opens up a whole new area as well. So it is a huge opportunity for the future, but not to replace taxonomists.
Great. Well, I think this sort of echoes John's earlier comment on the value of people as well as the value of science. I think that's all the time that we've got now for questions for this theme. So John, can I just ask you to wrap up for this theme and thank all the speakers and everybody for your questions?
Yeah, I mean, Chris, Alistair, Tony, and Marianne, thank you so much. Not just for your talks, but your insight. I think that's the thing. I mean, and almost anyone can give a talk if they practice, but the insight that comes from talks is absolutely invaluable. And that's a very human thing, even in science. And I thank you that each of the talks, very human as well as scientific. Thank you.
Lovely, thank you, John. We now have a 10-minute break. So if you'd like to go and refill your coffee cups, and then we will restart again at 10 o'clock. So I'm just checking with everybody behind the scene that's okay. I think it is 10 o'clock, is that right? So we look forward to joining you again shortly and see you at 12 o'clock. Sorry, yeah, 12 o'clock, thank you. Did I say 10? 12.
All right. Yes, a brief quarter-hour break. Yes.